libo.
Global health officials are monitoring an outbreak of bacterial pneumonia in the U.S. that made its way from China and Europe.
Infections by Mycoplasma pneumoniae, a bacterium that causes walking pneumonia, are reportedly common in school-aged children and could be resistant to some antibiotics.
There's a global concern that there could be another pandemic.
In late 2023, kids were getting sick with bacteria that caused pneumonia. The infections were hard to treat because the bacteria were resistant to first-line antibiotics. In this case, the infection wasn't highly contagious, so its spread was contained. But in the long run, this problem won't look like a rapid wildfire. It will look like a slow meltdown in our health outcomes. We're witnessing the end of an era that began 100 years ago when Alexander Fleming discovered penicillin. Antibiotics meant people were no longer dying from cuts, postpartum infections, or pneumonia, but bacteria have adapted, and new antibiotic-resistant microbes are spreading across the world. Soon we'll see more people dying from routine dental procedures, joint replacements, or abortions. The WHO predicts that by 2050, resistant infections will kill more people than cancer. This field is drastically underfunded. It's not profitable for drug makers to invest in antimicrobial medicines, but some scientists are finding a new way forward by looking to the ancient past. You're listening to The Naked Mole Rat, a podcast from Libo Studio. This episode is about a project that sounds like science fiction, but it could save humanity from superbugs. My name is Ilya Kolmanovsky. Hello.
My name is Cesar de la Fuente. I work at the University of Pennsylvania, where I lead the machine biology group, or the De La Fuente lab. And our mission statement is to combine human and machine intelligence to unleash the power of biology, which we think will accelerate scientific and medical discovery.
Cesar de la Fuente started thinking about superbugs after his team did a study. They looked for the worst-funded medical research areas that affect the most people. At the top of the list were drug-resistant infections.
It's a little surprising that there's so few people, actively working in this area, but we think it's going to be huge to try to improve the world now and into the future.
Cesar says resistant bacteria already kill 1.2 million people a year. That means 45 people will die in the 20 minutes it takes you to listen to this episode. What do we do? Cesar and his postdoc trained a neural network to analyze molecules with antimicrobial properties. They then used that data to look for new kinds of antibiotics that those bacteria could not resist. Not now and not ever. But where to look? Historically, scientists have discovered antimicrobial substances in fungi and plants. But these scientists are mining an unusual source, the genomes of mammals, specifically extinct mammals. Even more specifically, extinct humans. This all stems from a recent discovery that humans have something called antimicrobial peptides. These are strings of amino acids. They break off from larger proteins and they're deployed like patrol boats from warships to intercept invaders. For example, breast milk contains a large protein called lactoferrin. So when an infant drinks breast milk, some harmful microbes can potentially enter her stomach. But the baby's own digestive enzymes can break a piece off from lactoferrin. And that very piece transforms into an antimicrobial agent that lines the infant's stomach walls and protects the infant. Humans are full of proteins that have peptides like this within their structures. These fragments could be cut out with targeted enzymes. Do that, and you could unleash a fleet of bacteria-fighting peptides.
And so we have a couple of ideas for how to do this in mice, and it's within the plans that we have for the near future. But the vision that we have is that sort of the model is that a lot of these fragments are getting released all around our body. And we also know through experiments that they potentiate each other's activity to target pathogens. So they work together. And then I think that they're all around our body and they're contributing to host immunity. And they were previously sort of unrecognized effectors of the immune system.
20 years ago, scientists would have begun this slow process of studying all this in real organisms. But in the 21st century, we're skipping that stage. Artificial intelligence can analyze all existing human proteins, that is, the complete human proteome. It can search the enormous labyrinth of molecules for those small fragments that might work as antimicrobial drugs. Let's say it works. Let's say we can identify antibiotic peptides in human proteins, then use those peptides to make new drugs. We still might have a problem. They might not work. Modern microbes might already be familiar with our peptides. We've got to widen the search, and this is where things take a really unexpected turn.
And then so we started wondering whether we could find similar sequences all throughout evolution, not only in Homo sapiens or in modern humans, but all throughout evolution. And that's what led us to starting to explore the proteomes of our closest relatives. And these are Neanderthals and Denisovans. And so using available proteomic data, we explored some of those sequences and we were able to find potential antibiotics encoded in archaic humans. And what we then did was fascinating. Using chemistry in the laboratory, we literally resurrected those molecules that are no longer expressed in living organisms. And we resurrected them in the laboratory and we tested them against bacteria.
This idea that extinct humans are a treasure trove of medical information is very new. It started with a Swedish researcher in Germany named Svante Pääbo. 20 years ago, he figured out how to extract and decode DNA from ancient bones. That was the start of a totally new era in archaeology, and he was always praised for that. For example, thanks to his method, a whole new species of humans, Denisovans, were discovered. Their DNA was extracted from just 2 teeth and a tiny bone of a girl who lived thousands of years ago in a cave in Siberia. Then, scientists also discovered that we, Homo sapiens, interbred with Neanderthals and Denisovans. Looking into our ancestors' dirty doings was all very interesting, but then a pandemic came and it brought a very unexpected medical discovery. When Pääbo's group made this discovery, I called him up.
So my name is Svante Pääbo, and I'm a molecular geneticist at work at the Max Planck Institute for Evolutionary Anthropology in Leipzig in Germany. Well, so one of the people in our lab, Hugo Seberg, who was involved in a big international consortium to study the genetic predisposition to becoming severely ill when you're infected with SARS-CoV-2. And we were extremely shocked in June last year when we then found that this locus on chromosome 3 that is associated with increased risk to become severely ill with COVID actually comes from Neanderthals, the risk variant, the one that predisposes you to be more likely to become severely ill if you're infected.
So that was bad news. Pääbo called this the revenge of the Neanderthals. He told me that Neanderthal genes killed 200,000 humans during the pandemic. Studying those genes could help us find treatments, and not just for the carriers of those genes, but for everyone. But then came an even more interesting discovery.
The protective variant from Neanderthals on chromosome 12 reduces your risk with about 20%. So the effect is about 5 times smaller. But there it's the same thing. That variant is quite frequent and actually everywhere outside Africa, varying between 30 to 50% of people carry this variant in Europe, Asia, and so on.
So that's probably something good.
Yes. And in that case, one actually understands the mechanism. This fragment, the Neanderthal version, encompasses 3 genes that are well known to be involved in degrading double-stranded RNA. And the viral genome is, of course, made up of RNA, so this is a well-known mechanism to defend against RNA virus infections.
Neanderthals had a gene which codes for an enzyme. That enzyme attacks viral RNA. Some of us inherited it from flings our ancestors had with Neanderthals, but could that process become the basis for an antiviral drug? Paabo explained this on a Russian version of our podcast, and the Nobel Committee must have been listening. They awarded him the Nobel Prize in Physiology or Medicine that same year. Over the past 200 years, archaeologists have unearthed many ancient human remains. Over the past 20 years, other scientists have extracted hundreds of extinct human genomes from these bones. So now, virtual expeditions are mining those genomes for cures to modern ailments. Of course, all of this might still turn out to be complete nonsense, an AI hallucination, until we conduct a wet experiment. Here's what César de la Fuente told me about it.
So we just started thinking about this, and actually, it's pretty funny because we were having a lot of brainstorming sessions with the team, and immediately the concept that came to mind was the concept of de-extinction and the concept of sort of Jurassic Park, right? A movie that at least everybody in my lab, we all enjoyed when we watched it growing up. And it's the concept of bringing back to life entire organisms. But of course, that has a lot of limitations, including ethical and ecological and frankly technical limitations. I actually don't think it's possible to bring back entire organisms because we don't have enough genomic coverage. But what if we could bring back molecules from the past that no longer exist that could help us address some of the pressing issues that we have today?
Do you remember that meeting or that conversation?
Yeah, absolutely. It was a number of meetings, and I actually proposed the concept of molecular de-extinction, and I said, guys, why don't we try this? Bringing back a dinosaur has a lot of issues associated with it, but bringing back a molecule would be a lot easier in terms of sort of ethical issues and other things.
I'm at the University of Pennsylvania. I came here to see the lab where Neanderthal peptides are being resurrected. We're going to the lab of Marcelo Derterosian, who works with César de la Fuente. I ask how he's doing. He says he's tired with all the experiments he's running. He's got mice with infections, petri dishes of bacteria, and here we are, test tubes of peptides.
Does it feel anyhow special when you hold a test tube and this is something that
definitely nobody else ever synthesized that or nobody else ever saw that? Yeah, that's very cool. It was very interesting.
And then
it was a kind of a unique moment.
And then what do you do with it?
I tested it. I was so curious. They didn't last for 1 hour on my desk. I just got them all. I weighed them. I prepared all the stock solutions for everything. Next day, we were trying against all the bugs from the lab, including the multi-resistant ones. The ones.
How do you do that? How do you test them?
So you basically just grow the bacteria. You get them into a broth. So you grow them in agar. You get them into broth, you let them grow, let them get stronger and everything. So they are in an infection. I'm a chemist, as I said, so I didn't have any experience with bacteria before I started working with antimicrobial peptides.
AMPs, antimicrobial peptides, are those short and powerful molecules that could be cleaved off from large proteins.
So it was always weird to be holding a vial with a lot of deadly bugs inside. So it's let's be careful here. Then we just expose them and wait to see how active they were.
Next, he synthesizes the Neanderthal peptides that the AI found and observes how they interact with the bacteria in real life.
Yeah, we can go downstairs to the desk space. Is more comfortable. So this is the pathogens room, so we don't usually go.
He takes me to the room where he keeps the most dangerous bacteria. Then he runs out to grab a test tube.
I'll wait here.
I'm alone in a room with drug-resistant bacteria. I feel relieved when Marcelo returns with a tube of resurrected peptides.
And where do you keep peptides?
The peptides are all in the freezer, so they last longer. But they look like a powder, fluffy. So
I have to see that.
So here we have all the stocks for the 3,000 peptides that we are currently studying. So they are all.
I'm now holding test tubes with the resurrected molecules. They look like a white powder. That's what they sprinkle on the bacteria. Then comes the moment of truth. We need to infect real mice with those terrifying pathogens. And then we'll try to save them.
Tell us about any mice experiments.
Yeah, so for mouse experiments, we do the same. We grow the bacteria, we let it grow stronger and everything. We wash them with buffer. When it's ready to go to the back of the mouse, we just shave the mice. We perform like an abrasion on the skin, and we infect that, like simulating like a wound, a topical wound. And then we let the bacteria grow, form a biofilm, a nasty abscess, and then we just treat the abscess with the solution of the peptide.
Do you remember the first time you tried that with those?
Yeah.
peptides?
So we tested them against many mice because we wanted to ensure that we're having like reliable hits. So it was a long experiment. It took me like the whole day to do the infections and treatments. So it was approximately like 12 hours doing that, like surgeries in the back of the mice and infecting them, and then later treating all of them.
So they make scratches on the backs of the mice and introduce the deadly bacteria. Then they treat half of them with Neanderthal peptides and leave the other half untreated. The untreated half die from these scratches within the first 48 hours. And what about those we treated?
So what happens?
Yeah.
What happens?
They clear the infection, some of them. So this is very cool because especially the ones from Neanderthals, they cleared the infection very well, like 2 of them, right? It was amazing to see like a Neanderthal peptide killing like modern bacteria. Very cool.
What does it tell us about Neanderthals?
I don't know. We're trying to answer that. I want to, at some point in my career, I want to find like an antibiotic that's effective against all those nasty, like super bugs.
Are you getting there?
Yeah, we're going to get there.
I mean, this looks very promising.
Marcelo says their research is not limited to ancient humans. They're looking for these peptides throughout the entire tree of life, across all current and extinct organisms. They found tens of thousands. So far, only 9 have been actually produced and tested on mice. They found tens of thousands. So far, only 9 have been actually produced and tested on mice. Will we one day be able to buy Neanderthal peptides at a pharmacy to treat an infection? For this to reach the public on the open market, it needs to be profitable. To be profitable, it probably has to be patented. So here's a question. Can one patent a Neanderthal gene if it was created by nature?
Can you patent a Neanderthal molecule?
Yeah, in terms of patenting, so I've engaged in conversations with the patent office here
What are they telling
at
you?
the university, and they don't know, right? Natural sequences are not patentable, but sequences that were natural in the past but are no longer expressed in living organisms, it represents a new area of patent law. And I'm writing an opinion piece with a patent lawyer, to try to provide an opinion on this topic. Again, this is something that has not come up before in science or in patent law, and so there are many ongoing conversations to try to determine whether these sequences are patentable or not. So it's a very exciting, actually, new emerging sort of area of thinking. And again, we take that very seriously, both from a bioethics perspective and also just wondering, are these things patentable or not?
But that's not the only issue. Resurrection of peptides will require an ethical discussion. It might even be more fraught than the debate over whether or not to bring back the woolly mammoth, because such peptides might worsen the very problem they're intended to overcome. 100 years ago, we got antibiotics from fungi, and over the last 100 years, they have become less and less effective. These short peptides, even if we obtain them from Neanderthals, are much closer to our natural defense mechanisms. And this means, if we flood our bodies with these peptides in drug form, bacteria will get more opportunities to adapt to and bypass them. If microbes learn to overcome those medicines, they'll also learn to overcome our own natural defenses. That could become catastrophic. Any infection by any bacteria could turn into a very severe illness. So what do we choose? Protection for specific people right now, or safety for future generations? In the short term anyway, we might at least hope that experiments by scientists like Cesar de la Fuente lead to treatments before resistant bacteria get around to killing us. This is The Naked Mole-Rat, a podcast from Libo Studio. It is produced by Andrey Borzenko, Ilya Marritz, Danil Astapov, Anastasia Medvedeva, and Lika Kremer. Our sound editors are Alexei, Vorobyov, and Ildar Fattakhov. And I'm Ilya Kolmanovsky. Goodbye.